So when I sat and did my slides last week, I got quite nostalgic because it's almost on the day, 20 years ago, since I started my master's. So I'm going to try to outline for you a little bit about my travel, I will say, in the world of science and research and why I'm doing science. For me, every good research project I start with a good question, why? So why am I in science? So I come from a pedigree with not only strong female role models. This is my grandmother, who was born 1898 in Prague, in then the Austrian-Hungarian Reich. And she was the first female to have a DPhil in a STEM research field then, and she wasn't even let into the classroom. She and the Jewish boys had to stand outside and take notes looking through a glass window. So her degree and her commitment to her research, and she then went on to do a career in chemical research, has sort of really inspired me. The other reason, which is more personal, why I'm in science, is that a lot of people in my family has been afflicted by various common complex traits. And this is my mother and my brother, and I've lost both of them to cancer. And I very early on got very interested in why some people get sick. I have asked that even since I was little, and I decided that I was going to become a medic. 3 words that my mum has taught me that I've held very close to my heart and I carry with me in my work ethos and everything I do is polite persistence and perseverance. I think to succeed as a scientist today, especially in the large-scale collaborative environment we're in, you have to be kind. You have to want to work with other people and understand the differences, and you have to persist and persevere. So that's why I'm in science initially. When I was going to start medical school, my parents had an intervention with me and told me I was too mushy and that I was much more mathematically inclined, so could I please think about something else to do, which startled me a little bit. So I started to do a maths degree at university, so I spent almost a full year doing maths and I just loved it. And then I was told by one of the professors that it was better if I thought about something more applied to do, because mathematics didn't really have any career trajectory, which is unusual career advice. But I then soul searched and found out that genetics and biology was my second big love. And genetics has a big statistical component, so it suited my sort of personality, if you want. So 1999, I was really lucky to get introduced to this man. It's Leif Group. Throughout my talk, I'm going to acknowledge some of my mentors that have meant a lot to me. So Leif is my scientific grandfather, and he's a wonderful researcher, for those of you who know him. And he's been a very dear friend of mine throughout my career. So we started out looking at common complex traits, traits that have a genetic component, but also an environmental component. And there are many different genetic variants acting in concert with the environment to create the disease. So my project per se was looking at type 2 diabetes in really large scale pedigrees from Finland, where diabetes was really sort of, it looked almost like an autosomal dominant disease, and it was clearly sort of segregating in the family. And the big en vogue at the time was that you could genotype microsatellites, which are di, tri, or quadrinucleotide repeats throughout the genome. Augie's nodding because we've been collaborating with Decode ever since. So I've known Augie for a while as well. And then what you do is basically trace the parts of the genome that is the co-inherited with the disease, but not with the people that are not ill. And then you use statistical genetics and calculate if this is identical by descent or identical by state. And you, if you're lucky, these are the markers on the x-axis, and this is the LOD score, or the variance component, on the y-axis. And here is the first finding that I was involved in on chromosome 9 for type 2 diabetes, and it's sort of suggestive, almost significant, and we were super excited. But we were all hunting under the false premise. We had not yet understood at this time what the power inclinations were of the studies we were doing, because we had too small sample sizes. And we didn't really find anything significant out of it, if we're self-critical in our sort of hindsight. So then I started to work with Joel Hirschhorn. This is now being in sort of Lund in Sweden, but collaborating very closely with a group at the Whitehead Institute, the Genome Institute at Whitehead. And we started to think, but what's the classical inherited trait that we could look at? A model trait, if you want, that is a common complex trait, but has a clearer inheritance pattern than type 2 diabetes. And we thought that stature is a pretty good example. So what we did was to do a quantitative linkage analysis. You use a slightly different method, it's a variance component method, and we found 4 regions that were screamingly significant for that. And we felt very encouraged that this is sort of an example of how you go about pinpointing, and that we hadn't quite understood that we're underpowered in type 2 diabetes, because we had many more individuals that were informative for height. During this time, as I mentioned, I'd started to work with the Genome Institute, and that later became the Broad Institute. So I was very lucky because Leif very kindly taught me the power of collaboration. So he teamed me up with David Altschuler, who was a postdoc with 3 papers at the time on C. elegans, and didn't know anything about human genetics, and Mike Daly, who was a physics major but didn't even have a PhD, but they're obviously fantastic researchers. And they sort of coached me from afar, so I started to go back and forth. I'm not very good at jet lag, so they invited me to come over there for a year and a half to spend with them. And they set out 2 problems for me. And the first one was to really scale up association studies. So when you do an association study, you basically take a genetic variant, you look at cases, and you compare the allele frequency between the cases and controls. Or if it's a quantitative trait, you check the increase of the trait variance between each unit of the increase of the trait, and ask if there is an association with that. So the field of association studies has been plagued again with underpowered studies, inconsistent results, and we started to think, based on the linkage studies, that maybe this is underpowered again. So how do we go about this? And in parallel, global expression studies, the sort of gene expression arrays, made their debut. So they set out 2 projects for me to work on. And the first one was to look at 16 variants that had been reproducibly associated with type 2 diabetes. And this is the forest plot of one of those variants, the Pro12-ALA variant, and its association with diabetes. It had initially been reported in Nature Genetics, and then other studies have sort of said that it wasn't associated. But Mark and I sat and thought about it, and everyone who knows me knows I love a good confidence interval because it gives you a sense of where you are on the spectrum. So Mark said, why don't we just do a forest plot? This wasn't very often done in the field. By everything that has ever been published, and we marry with our data, and we see what happens. And we saw a consistent effect amongst all the studies. It's quite heterogeneous based on the phenotype and also the sample size. But it's actually the first or the second, depending on if you're Mark and Anna or if you're David and me, the first and second robust association with type 2 diabetes that we had ever put forward. And the lessons learned again is that we need to scale up power, and that association studies are probably going to be easier and much cheaper to do and much faster to do than linkage analyses. Another thing that we noticed is that we're probably not going to see massive effect sizes, because we started to think about effect sizes of variants here. And there was this appreciation that we thought that we would find the gene that sort of explained obesity, or the gene that explained type 2 diabetes. But here is where it really dawned on us that that's not what the genetic architecture is going to look at. And then what appears to be heterogeneity can actually be supportive across different populations if you take the confidence intervals into account. The second task they put upon me was to perform gene expression analyses in human skeletal muscle of type 2 diabetes patients, patients with impaired glucose tolerance, and also healthy controls. The sample size numbers look laughable today, but this was one of the biggest gene expression studies performed at the time. And I got stuck in American customs for importing human flesh, and won't tell you that story. I will say that for the champagne, otherwise I'll draw over time wildly. So when we came to the Broad, they basically had a collaboration with one of the companies performing these arrays, and they had a fridge where you could just go in and take them, and they were like thousands of dollars each. So for me, that was almost like Christmas. And so I did all the experiments, and then I worked together with Joseph Lihar, Pablo Tamayo, and Vamsi Mota. And what we found in the beginning was that if we just do a case control comparison, there wasn't very much there. So we developed a gene set enrichment strategy for looking at the data, where you collect the gene sets based on a randomly pre-annotated notion of how genes interact with each other. So it could be Go terms or KEGG terms or whatever your preference is. Then we order genes by their expression difference. Then we measure enrichment for each gene set. And then we record it for the actual data. And then we permute it a couple of times. I think 1,000 times was too little, so we actually ended up with more than that in the end. And then you basically get a distribution of your permuted data over your observed data, and you can see if it's significant or not. And what we found is that if we compare type 2 diabetes with normal glucose-tolerant individuals, we see that the genes that are involved in the oxidative phosphorylation pathway is coordinately downregulated in human diabetes. And we also, with Ewan Rosen's help, who's a molecular biological god at Harvard, we saw that all of the genes, or most of the genes, are actually PGC1 alpha responsive genes. So this was a big breakthrough. Some of the critics in the field will say that some of this was known before, but that's mainly from urine studies, which doesn't disregard it, but this was the first time we saw it in humans. So then I came back to Sweden and I defended my thesis, and here I have a photo of Lina Peltonen, who was my thesis examiner, and she became my mentor. Lina has meant a lot to me because she's a female role model who has a family, who's been wildly successful in the field. Tough as nails, we didn't always agree on the science, but she was fearless, and I really, really admire her for that. So she has meant a lot to me for my career. So after I came back to Sweden, I was a little bit in limbo and didn't know exactly what to do. And during this time also, the Human Genome Project had finished. The SNP consortium had shown us that there was a common SNP at least every 300 base pair throughout the genome. Then there was the HapMap consortia, which Gil and many other people who were really sort of important to lead, that showed us that the SNPs didn't exist in sort of a vacuum, but had relationships with each other, which we call linkage disequilibrium, which tells you how closely you can predict one SNP based on the information you have on another one nearby. And that led to the realization that first we had single nucleotide polymorphisms, which are largely biallelic. And for anyone who works in computer science, you will know that sort of binary input to computers makes it easier to work with. And in parallel, the linkage disequilibrium and the fact that SNPs travel in big blocks together, and that you don't have to genotype all the SNPs in a block in order to obtain the information, led to the dawn of new global genotyping arrays that are designed so that you can genotype about 350,000 markers instead of 11 million. So that will save you time and money and cost. And suddenly we could start to think about scanning the entire genome for association studies, and genome-wide association studies arose. So here I got really excited. So I was at American Society of Human Genetics. I was in a chocolate martini bar with Mark McCarthy and Lon Cardon, and we played poker. And I was a postdoc in Sweden at Karolinska Institute at the time, but they started to talk about the Wellcome Trust Case Control Consortium, and I had heard about it, and it was super cool. And why was I in Sweden? I could come and join them because they needed people who could do analyses. And I got super excited because I've always been one to rise to a challenge. So I managed to secure a fellowship, and I came there to work with them. Lon very quickly left to go to Fred Hutchins. Mark stayed, and I managed to get a Wellcome Trust Career Develop Fellowship, which was exciting. Peter Donnelly came, and he took over at the Wellcome Trust Center directorship. And Peter is another person who has actually been a wonderful mentor to me, and I have a lot to thank him for. He's a Women in Science champion, for those of you who doesn't know it, and he's always been very supportive of me. So one dark December night, I sat with a big hat on a yoga ball next to Jeff Barrett, who was working on another trait, and Nick Timson, who was working with me on the type 2 diabetes thing, and the data for the Case Control Consortium came in. And Gil just asked me when we interviewed what my biggest eureka moment was, and it's actually this Manhattan plot, because this is the first Manhattan plot I have ever generated. And Mike Whedon, who is in the audience today, was on the phone with the Exeter team who was running the data in parallel so that we can cross-compare to make sure that we didn't do any errors, because this was analysis tools that we hadn't really deployed before. So basically, it was done using a 500K SNP chip, and in about 2,000 cases and 7,000 shared controls. And the first chimney that came out, Augie will smile, because that had already been reported as an association from the DECODE group that they had found through association analysis of a microsatellite marker, incidentally. And then the second chimney, which is a sort of an association peak, so on the y-axis here you have the negative log 10 p-value for association, and on the x-axis is chromosome and position. And for the pedantic person in the audience, you will say, well, that's not really a genome-wide association, it's an autosomal-wide association, and I think that's true. But for ease of discussion, I'm going to call it a genome-wide association study. So we found FTO, and then we really excitedly called up our friends in Boston and in Michigan who were working on similar projects but in different data sets, and we're like, oh, this is what we see, what do you see? And they did not see FTO, and we got really disappointed, and we thought that we're actually wrong here. And then on Christmas morning, Mark emailed me and Mike Weedon, and he said, oh, it's going to be because our cases are overweight or obese, and the other ones are working on lean Finnish cases. So can you please just look if it's associated with BMI? And then the race started. So I guess my Christmas and your Christmas, Mike, was destroyed by this, but we were excited to analyze data. So there were a few realizations here. So we needed over 77,000 individuals to find this association. The per risk allele is about 30% increased risk for obesity, which translates to about 1 to 1.5 kilo increase in weight. So for those of you who think that all common variants have small effects, I would counter-argue and say this is not a small effect for a common variant, actually, that you can differ from your neighbor in your neighboring seat by 1 kilogram, depending on, or even 2 kilograms, depending on your allelic makeup at this one marker. The association is present at the age of 7 already and reflects a specific increase in fat mass. So based on this realization, 77,000 individuals is beyond anything I had ever genotyped for association studies. The PPR gamma paper was, I think, 3,000 or 4,000 individuals, and that was a lot at the time. So we quickly realized that we need to aggregate together in consortia now, start to collaborate with people. So we called up our friends in Cambridge. We're like, we're done. Are you done? Do you want to pull your data? Yeah, let's pull the data. And we found one other hit, a locus 188 kilobases upstream of MC4R that was associated. Really interesting because MC4R is one of the key candidate genes for monogenic obesity. It's one of the first monogenic obesity genes that is reported. It's been reported to be disrupted in monogenic mouse models of obesity. It affects satiety, sensation of hunger, and energy metabolism in children with a very early onset. But the variant that had been reported with an allele frequency of 0.5% in the population didn't show any association in our data. So instead and this is a coding variant in the gene, but instead we saw this regulatory unit. And that's another realization we've had along the way, that most of these common associations are actually harbored in intragenic or sort of non-coding genes. And I'll get back to that. So then we realized that we had to expand even further, and the Genetic Investigation of Anthropometric Traits Consortia were founded. I'm excited to be on the steering committee, and I chair the fat distribution group within that consortia. So there's been a virtual explosion of gene discovery. So this, I hope, looks almost like an exponential or like a bombastic sort of increase in identification, where I talk to you about the FTO and the MC4R, and then there's a virtual fruit salad of gene names here. I want to issue a word of caution because most causative genes are not determined yet, so we're a bit sloppy when we sort of adding gene labels to it. We need to be careful with that. So I would discourage anyone from jumping out in the lab and start to work on it, because we have a lot of proving of what's actually causal before we continue. So I'm just going to talk through some of the lessons we have learned so far. So the first one was when we looked at the fat distribution data. So this is just the association p-value mapped against the heterogeneity p-value. So one of the key challenges working with big data is heterogeneity, non-randomness in missingness of data, or some sort of consistent latent variable that you don't know what it is that is sort of biasing your data. So cleaning is incredibly important. So Joshua Randall, who was a student with me at the time, and we started to look at the heterogeneity quite extensively. And one of the things we noticed is that roughly here is where your robust associations, you would want them to be because they have non-significant heterogeneity, but you have significant association. And we saw these sort of flying clusters out here, and I got really worried because I'm quite anal about QC, as people who works with me know, so I'd like to test run things for other people and so forth. And we couldn't quite figure out what it was. And then we thought about it a lot. And I said to Josh suddenly, like, I bet you that this is going to be driven by the difference in men and women. Because if you run the genome-wide association analyses in females versus males and do, this is called the Miami plot, where the male association is going down, the negative log 10 p-value. And for females, it goes up, the negative log 10 p-value, and you can see almost like a mirror image of each other. You will see that the association in women on the autosomes is much stronger than that in men. And we noticed that for about 50% of our loci, there is a significant difference in the effect sizes between men and women. And if we compare that with the overall obesity work, or BMI, it doesn't look like that at all. It's about 3% of the loci. So there's a really stark difference there. And it turns out that if we look into those loci that were these sort of heterogeneity outliers, those are the ones with the strongest differences in betas. And then about here, my husband got recruited to go to Boston and do a postdoc, so I had to follow. So I went again as a scholar in residence, which I don't think nobody ever knows what that title meant, but I had a lot of fun. So we continued to do a lot of work, and we now started to prod on the pathway analyses of fat distribution and overall obesity. And the person I show here is Tune Pash, who is an associate professor in Denmark now, but was working with Joel Hirschon at the time, and he and Lude Franke had come up with a method called DEPICT. And what DEPICT does is to give you the most likely causal gene, the sort of associated pathways, and then an enrichment for cell types and tissue types in a statistical framework. And here I'm just showing you the pathway analysis of fat distribution, which basically mainly implicates adipose biology and insulin resistance pathways. But also note the female infertility and the transcription factor pathways coming up significantly. In parallel, when we look at over 77,000 gene expression analysis data points that has been pulled out of GEO, when we ask the question, if you look at the associated loci, in which cell types and tissues are the genes in the associated loci expressed up or higher or lower than the background of the genome and in a permuted way? And the only tissue type and cell types that are coming up is adipocytes and adipose tissue. And this is in contrast with that of overall obesity, where we basically just see large clusters, almost like bee swarms, for neuroendocrine pathways and neuronal tissue. And here's the tissue. It's the same tissue and same experiment as for the fat distribution, but just the different GWAS input. And you can see the sort of really stark sort of enrichment in neuronal pathways. So it seems like fat distribution is more a peripherally driven phenotype, where overall obesity is a central neuronal sort of phenotype. This is a generalization, I should say, but there are other methods. Hilary Finucane has shown this with her LD score regression partitioning methods. We have seen it using other data that I'm going to show later. So we feel quite confident that this is sort of robust. Then I always get the question when I've sort of gone up for fellowship interviews and so forth, like, you work on anthropometric measures, it's not really sort of doing anything clinically. But I would want to remind you that both BMI and waist-hip ratio are used by WHO for definition of cardiometabolic risk. So they are clinically validated measures. And while we would be incredibly excited on having really large-scale scan data, because what we really want to access is, of course, the real fat distribution rather than measuring skeletal sort of structures in the hippie region or the large gluteal muscles. And that sort of is underway. So in UK Biobank, as everyone here will know, there's about 100,000 MRI scans underway. So in my team, we have sort of started to draw out plans and sort of think about how we're going to address the MRI scans using machine learning techniques, which we're very excited about. But in the meantime, I had the pleasure of working with Audrey Hsu and Caroline Fox, looking at 9,000 women and 8,000 men with MRI or CT scans, and look at 6 ectopic fat traits. And ectopic fat is when fat starts to shunt into places in the body where it's not supposed to be. So you can have fat in and around your kidneys, fat in and around your heart, in muscle, and so forth. So we found about 1 locus per trait we looked at, and that's a reflection of the very low power. And there is an important lesson to be learned there, because somebody told me that if you look at 9,000 people with precise scan measurements, you will outperform any of the anthropometric sort of data, which is obviously not the case. So I think we will have a win in terms of power, but it's not going to be on that order of magnitude. The second thing that is interesting is that there were a slew of functional experiments performed on some of these genes that were in the associated regions. And when we look at that data, it looks again like these ectopic fat traits are largely driven by adipocyte development and differentiation, which again ties in with what we found for the anthropometric traits, which is exciting to us. So then another question that we've been sort of thinking about is, well, you look at waist-hip ratio adjusted for BMI. So we usually adjust for BMI, and I will come back to that. We already use BMI in the clinic, so do we need another measure? So I think I've shown to you that we capture a different type of biology with it. So therewith, we think that we capture a different type of risk, an independent part of risk. Lindgren et al. Published in JAMA in 2017, looking at a polygenetic risk score for the fat distribution traits that we had sort of put forward, looking at type 2 diabetes both in cardiogram and in UK Biobank, the interim release, and also coronary heart disease and cardiogram, showing that waist-hip ratio has a strong causal pathway for both type 2 diabetes and cardiovascular disease that is independent of that has already been shown for BMI, which we think is sort of exciting, and we're taking that further. So Jenny in my group is looking at that at the moment, splitting out and looking at sexual dimorphism, for instance. So then hot off the press, so Sarah Pullit, who is a Veiney Fellow in Utrecht, and I have the great pleasure of collaborating with, have just bunked together UK Biobank and the giant data for fat distribution, and doubling our sample size, almost 10-folds our discovery, which is really exciting. So here we're looking at 27.4 million variants in about 710,000 individuals, and we find 469 independent signals compared to the 49 that we had previously in 346 loci. Note that we use a slightly more stringent significance threshold here, but that's to penalize ourselves for the larger number of variants and also for the fact that we're testing a lot of traits. So I talked a little bit about how we adjust for BMI before, and we do that because we basically want to look at fat distribution. If we assume everybody was the same height and weighed the same, what would the actual difference in the fat distribution be? So Hugh Achard, who's a really clever guy who used to work with Alke's Prize, saw one of my talks when I was in Boston, unfortunately. But then he put forward this concept of collider bias in genetic association studies, which has led to a lot of discussions for us. And it's basically if you have a quantitative trait as your outcome, and you put a correlated quantitative trait as a covariate in your model, you'll get an increase in sort of positive findings that might be type 1 error. So that's obviously quite serious if somebody puts that forward and you don't know quite how much. So it's something we've been thinking about and we wanted to address seriously. So Sarah has looked at this, and somewhere between 5 and 10% of our significant loci will be afflicted by collider bias or pleiotropy. Because if you read Alke's and Hugh's paper, and I've discussed this with Alke's a lot, you cannot distinguish between collider bias and increase in type 1 error or really closely shared biology. And for me, who has the biological discovery at the core of what I'm excited about, I think that pleiotropy is one of the biological phenomenon that I'm really excited about. So it's something we need to keep in mind. So now we're actually repeating a lot of the pathway analyses, including and excluding variants that have signatures of pleiotropy or collider bias, just to see how it sort of affects our interpretation. From what we've seen so far, we're not particularly worried about it, but we need to be aware and open with it because we're learning along the way. We should note that type 2 diabetes, for instance, is afflicted by collider bias of BMI per the nature of the trait. BMI has collider bias with height, for instance, by the nature of how the trait is derived, but a lot of people don't discuss it very openly, and I feel that we as a community should be excited to learn moving forward. So then we have realized how low heritability and how low the effect size are for the common variants, and Terry Manoli and colleagues had sort of put this figure out, and I'm certain a lot of you will have seen it. So the common variants will often be associated with low to modest allele frequency. And then there is the case of the missing heritability. So it became a big discussion in the field. People were discussing why didn't we explain more of the heritability. We haven't really assayed the low frequency variants with an intermediate effect or even the rare alleles causing Mendelian or even personalized sort of penetrant variants due to the way that the experiments are designed. So here, Gil and I had a discussion, and Gil convinced me to come back to the Big Data Institute, and we started to work on the giant exome chip effort, where we look at about 500,000 individuals for waist-hip ratio and about 720,000 individuals for BMI. We look at about 225,000 markers with a minor allele frequency of 1 or lower, and we do a slew of analyses. And this was accepted in Nature Genetics last week, which I'm really excited about. So this is just an analysis result from a single variant test. And you will see that we have found 28 novel variants, but only 9 of them are novel and rare or low frequency. And that's in 420 individuals. And I would say that that's a quite low yield. This is a chip that is particularly designed for this in quite large sample sizes. So that was surprising to me, at least. We see variants that have a larger effect size, but not brutally much larger. I think it's about 3-4 times larger, but not sort of standing out in a stark way. So then the sexual dimorphism signature is also present in the fat distribution effort, but not in overall obesity, so that's replicated here. And in the interest of time, I'm not going to go through it in greater detail. So we also had a paper in Nature Genetics earlier this year looking at the overall obesity here, where again we find 9 variants. This is in 720,000 individuals, where 9 of the variants had a minor allele frequency of less than 5%, which is not astonishing by any means, I think. We do learn novel biology, and there is a lesson to be learned here, and I think looking at the common variants is going to continue to be exciting. So I just want to give you one nugget from this paper, and that's our favorite MC4R, which I talked about before, where there are 2 variants that have a minor allele frequency of about 0.1%, which means that there is about 6,400 carriers for each of those. And they carry a per allele effect of about 7.5 kilograms, and that's quite whopping. So for those of you who think that it's just to go out and run, and just to eat a little bit less to maintain your body weight, you should appreciate that if you have the wrong genetic makeup, you're just going to work that much harder, and it's going to be that more difficult. So if you're naturally slender, you're not going to think about it. But it is really important to think about. If we look at all the common variants aggregated together, that's another, the polygenic risk carries about between 10 and 15 kilograms, depending on how you calculate. So suddenly that's a quite big impact, actually. And we need to think about it, I feel. I thought it was particularly interested in the correlation with a study that I just read from Karin Clement's group. And Martin is going to get chills when I talk about this, because it's a clinical trial with an N of 2, and it's not randomized or controlled in any way. But these are 2 individuals with a genetic POMC deficiency, so they have mutations in the same pathways as MC4R, for instance, the MC4R mutations. And they are treated with a melanocortin 4 receptor agonist called setmelanotide. I have no commercial interest in any companies that are producing these drugs, but that is already on the market. So they were thinking that, okay, so here we have a drug that is targeting a pathway that we're now seeing more and more evidence that it's crucial for weight control and weight maintenance. How about we take those individuals who are challenged here and give them setmelanotide and see what happens? Note that it's an N of 2. But within 42 weeks, the first patient had lost 16% of their body weight, or 51 kilograms, with no other treatment recommended. And the second one had lost 13% of the body weight after 12 weeks. So the company that now is producing setmelanotide has sort of noticed the work that we've been doing, and they want to sort of figure out and find patients with these mutations to see if they can sort of expand that out. And the reason I'm showing you this is just that it's an example for how the biological knowledge can actually be translated into repurposing of drugs, or finding people that would be particularly susceptible for effects of drugs, for instance, which is core of what we're hoping to do with our research. So I think the key mechanisms in overall obesity and fat distribution that my research and that of my collaborators have put forward is that overall obesity is a trait or a condition or a disease, even depending on where you sit on that fence, that seems to be regulated at least in part by the central neuroendocrine control of energy balance, where that of fat distribution seems to be regulated at least in part by much more peripheral mechanisms, including adipose tissue. And I want to point out as well that for those of you who think about the underlying biology of fat distribution, that in the development of the mesoderm, and you have mesenchyme differentiation, bone, cartilage, connective tissue, and adipose tissue. So bone, skeletal muscle, and adipose tissue sit on a joint developmental pathway. So when we see bone pathways coming up for waist-hip ratio, it does not necessarily mean that we have targeted a bone pathway. It might actually mean that we're looking at mesenchyme differentiation, and that's something we're thinking about in my group. So I'm going to spend my last minutes, I can see what time it is, but Gil, you have to start a wave, on thinking about future direction and ongoing work in my group. So I'm first going to give you a summary and then give you a couple of short vignettes. So currently in Giant, we're aiming to scale up to 7.5 million individuals with genetic data and obesity phenotypes in the coming years. That sounds a bit absurd, doesn't it? So in the way we're working on now, where China Kadoorie Biobank is coming in and a couple of other biobanks have joined in, I think we're on 3.5 million genotypes, but there is a lot of big efforts going around the world, which is really exciting. I am personally really excited, especially being here at the BDI, in expanding the range of phenotypes and mining electronic health records for genetic discoveries related to this. I'm excited to work with MRI data. I'm excited to work with histology data, and I'll talk more about that later. So in my group, we're also working and thinking about machine learning to learn more about the phenotypes and correlations, to identify patient clusters and stratifications, and also prediction of outcome models. And we're working together with the Harwell team to do deep comparison between mice and man, because I'm interested in why it is that we always dismiss it when we often note that the comparison have a lot to offer. We also work on dissecting causality versus correlation with a slew of comorbidities. And I'm interested in large-scale, well-planned sort of biological work to sort of search for functional mechanisms. And I'm doing that in collaboration, for instance, with Melina Klausnitzer at the Broad. So I just talked about the sort of scaling up the genetics, and I just wanted to highlight and mention all my collaborative friends in a slew of consortia, but mainly the PCOS consortia and the Giant consortia and the UK Biobank Cardiometabolic Trait Obesity consortia. And I also have a long-standing collaboration with a range of various biobanks across the world, where I know people and we have collaborated with them, and I think it's an exciting. I should add the Jerusalem birth cohort, because I have Hargit in my group now. So I think we sort of need to scale up genetic discovery. I think there is much more to be found there, so that's one thing we're doing. Also working with Michael Holmes and Sanna Peters, and also with Jenny in my group and Mark McCarthy, we're sort of thinking about how to dissect causality from correlation. How are obesity traits and other traits correlated with each other, both genetically and epidemiologically, and what does it mean when they don't coincide? If something has a really strong epidemiological correlation but it's not causative, do we dismiss it? Andy talked about that in his talk earlier this morning, or is it interesting somehow? Are they causally associated with comorbidities? Maybe we can learn about pathways that we can target for more successful intervention, both for the sort of endophenotype in the middle and the disease outcome. So, but closest at my heart is homing in on function and mechanisms. Which variants are actually causal? We have to work with fine mapping, both sort of deploying more sort of refined statistical methods and also folding in various types of functional annotations. And we have to sort of think deeply about scraping in sort of publicly available data. There is tons of fantastic data out there that we have the opportunity to work on, and sort of marry that with the fine mapping to increase our biological understanding, and then sort of propel that over to translation, hopefully. Which effector transcripts and functional elements are exerting their effects on the trait that we're interested with? Are there pathways and molecular signatures that we can leverage therapeutically? I've mentioned a few. One of the insights we have gained is that effect size is not equal to biological relevance, and some of the genetic associations that we have found in PPR gamma and SERCR and so forth show us that even modest effect sizes in particular genes, especially if there is monogenic evidence as well for a perturbation of a particular gene correlated with a related trait, will increase our success here. So I'm just going to give you a quick vignette here on how we think that we might go about mapping out some of the underlying mechanisms. And this is Karen Mulkey, and I want to attribute part of the genesis of the hypothesis here to her, because we worked together really closely in the giant fat distribution consortia. So when we looked at where our fat distribution SNPs were associated, we started to notice that there seems to be a lot of transcription factors that are involved in adipogenesis in the associated loci. And I got really, really excited, because if you think about the sexual dimorphism, there aren't that many ways that can actually happen on an autosome. It could be due to imprinting, or it can be due to transcription factors that maybe interact with sex hormones, for instance. But otherwise, I can't think of a lot of other clever arguments that I think would hold up. So if somebody here does, please let me know, and I'd be excited to work with you. But Karen and I thought about that. And then I launched people in my group sort of thinking about it. So we started out doing fine mapping, and we saw that in about 71% of the loci, the credible set contain 10 or less variants. We should note that this is on a hapmite2 scaffold, which is not particularly dense. So there is a slight bias there, but it was a starting point. So this is now done in giant data. And you will see that in some cases, we go down to one variant. It doesn't necessarily mean that that one variant is the causal one. It just means that it's likely the causal or really closely related to or nearby the causal one. So when we overlay those 49 regions with the credible sets, with the ChIP-seq binding sites, there is 167 publicly available ChIP-seq data sets with binding sites, and ask, are any of our associations consistently correlated with these? So do we see an enrichment for binding sites? And this is now in fat distribution in giant in male and female, and overall obesity male and female. We've done it overall and all as well, but we see a similar pattern. We find one transcription factor that has an enrichment p-value of 10 to the minus 9, where the Bonferroni corrected p-value is 10 to the minus 4. And it's called ECH2. And I almost freaked out. So Gil asked me about Eureka. This is another Eureka moment. And the reason for that is that if you go back to slides here, is that ECH2 is a polycomb repressor of adipogenesis. So it's a good candidate for it. But obviously, this needs to be replicated and validated further. And this is work done by Cal Galton and Juan Torres and Tuchek Haderi. And then we were a little bit nervous, because there we had only looked at the 49 loci. So we wanted to take it out using another method called FGWAS, and do it sort of globally, and just compare sort of what we saw. And again, ECH2 comes out as a top candidate in males, but not in females. And FOSL2, which is actually a leptin binding receptor, comes out as a significant one in female if you look globally, which is really interesting. So we're currently working on replicating this in UK Biobank, and also doing functional validation, both with Samantha Lauber in my lab at Roger Cox at Harvard. So the last vignette, I'm on time, I'm so excited, is that of adipose tissue then. So almost all the work on the fat distribution data has pointed us to the importance of adipose tissue, and the hypothesis seems to sort of hold through in the first initial in silico bioinformatics follow-up. So for me, who's trained in an endocrinology clinic and sort of been excited about adipose tissue for a while, I got really excited about the notion of hypertrophy versus hyperplasia, where hyperplasia is a state in your adipose tissue where you have a high number of smaller cells that is more insulin sensitive and it's associated with a healthy cardiometabolic risk, compared to that of hypertrophy, where you have large cells but fewer of them. You have a lot of inflammation and low insulin sensitivity, and that's associated with high cardiometabolic risk. So one point I wanted to make as well is that we don't see any evidence based on genetic data for an inflammation signature in obesity. And that's been controversial in the field, but we don't see that. I think personally it's a secondary phenomenon. It doesn't discredit it particularly, but it just means that it's not a primary cause or a primary driver of it. So I just wanted to mention it because I saw it there. So one of the things I was interested in is whether we could see a signature of hyperplasia in people with a sort of positive, I shouldn't say positive because it's the wrong word, but a sort of a beneficial fat distribution health-wise versus that of a negative one or a sort of unbeneficial. So just at this time, Craig Glastonbury started in my lab, and we had started, Sarah Pullitt and I, to think about this, but he proposed the sort of project in parallel to look at adipose tissue histology, to basically do in silico phenotyping of cells. So with that, you take histology slides from a lot of individuals where you have genetic information, so they have GWAS data, they have gene expression data, they have phenotype data, and then you would have to have it in large enough sample sets so that you could do something with it robustly. So Craig, who's a keen sort of machine learning analyst and is sort of expanding his interest and his skills there, and I came up with an analysis outline largely driven by Craig. So we have applied deep learning or convolutional neuronal networks to automatically count cells in the histology slides. So first you have to generate the training data set. So we were 3 people scoring slides in parallel. It's easy to start with adipose tissues. I feel almost like we cheated because it's 90% adipocytes. It's not like going into the gut where you have tons of different cell types and you need to be a hyper specialist in order to do it. But we did reasonably well, I would say, with a few outliers, such as myself. And then the CNN is basically redundantly estimated cell count using density maps. And then we have applied it in the first instance to GTEx, where you have about 700 individuals that are informative for subcutaneous adipose tissue. And then I think it's 250 that have visceral adipose tissue or adipose tissue taken from the abdomen. So we have applied this, and I think we have counted over 20 million cells now, and we have a test error of 11 cells per image, so over 93% accuracy, which we think is good. And the cell count is significantly and negatively correlated with obesity and age. We see a significant correlation with type 2 diabetes that has been reported previously, but actually it's BMI dependent, so it doesn't seem to really hold up without that. And it's not confounded by ischemic time, it's the time since death, because these are post-mortem samples. So we did the GWAS for this, and we have some promising chimneys, but nothing reached genome-wide significance. So we're now expanding this. So we have obtained through collaboration with the Exeter group and with Melina Klausnisser's group. So we have over 1,200 histology slides that we're doing. So we have burdened ResComp somewhat with this analysis, but we're excited to sort of do the next round of analyses. And this is work that I don't think I would ever have thought about doing, actually, if I wouldn't have been here at the BDI. So I'm super excited about that, because I'm really sort of learning along the way. And it excites me after 20 years that I can get into new methods and sort of move forward. So I'm going to sort of tie up everything here, so stick on time. I know everybody wants to get to the champagne. I hope that you agree with me that we have generated a considerable catalogue of robustly associated common and low frequency variants for various aspects of obesity. I think that's fantastic. When I started, I don't think there was a robust association out there at all for any of these traits. So that's a big step forward. GWAS has often been criticized for being slow, but we should all remember PCSK9, for instance, that has gone incredibly fast from discovery into a therapeutic agent. So I think that it holds a lot of promise. We have learned a lot about biology, and we are on the verge of moving it forward. Another thing that we have seen in our work is that the credible set of variants when fine mapping, or the sort of tightest possible association region, predominantly map to non-coding sequence. I think there is a big lesson to be learned there. People often want it to be a sort of surface receptor for a cell because it's easy to drug, but that's not necessarily going to be what the biology is showing us, unfortunately. So we need to think about clever ways to sort of go around that. I think gene regulation is going to be incredibly important to understand better. I think we've learned so much new biology. I think that the fact that overall obesity is a neuroendocrine condition is something that people wouldn't quite have bought into even 10 years ago. The fact that fat distribution has a distinct component that adds independent cardiometabolic risk is incredibly important because it allows a different way of thinking about therapeutics. I think we're in such an exciting place at the moment. I remember one of my mentors said to me once that once all data becomes publicly available, the challenge is upon us to ask the best questions, and the smartest person is going to have the best paper. So on that challenge, I'm going to actually sort of wrap that up. And I'm going to accept the challenge, and if I sort of fail in impact factor now, I'll be embarrassed. But I do think that we're in such an exciting time where there is abundance of opportunity to sort of move forward. So last but not least, I want to make a comment on personal genetics. So because I am a woman and I'm in STEM, I often get the question, which makes me a bit sad, from junior female trainees, like, oh, but how do you do it? And is it possible to do it? And do you have kids? And I don't always talk about it, and my kids are not prominent on social media, but here they are. I just wanted to say that it's all about, and I hope I've sort of acknowledged people along the way that have supported me in various ways. It's all about finding people that support you, that believe in you, that make it possible for you to be whoever, whatever you are, and do it. So it's not a question about if, it's about how and when. Make your life happen when you have the opportunity to do so, and make your career follow that. So I've been incredibly lucky, but I also have the best partner who is incredibly supportive and very, very equal. So that's another thing that I got. I was being recruited to Sanger at one point in my career, and I told them that I didn't want to go. I was quite happy in Oxford, and I just met my husband at the time. And then Lena Peltonen, was head of human genetics there, said, I actually respect that decision because it's harder to find a good partner than it is to find a good job. And that turned out to be really good advice. And again, I want to thank my team. It's been such an incredibly sort of exciting time to sort of aggregate you together. That's been one of the biggest joy, actually, coming to BDI. 2 of 14 people in my team are already on independent fellowship, so mentorship is something I take incredibly serious, and I really, really enjoy. So I hope that all of you paid forward when you stand here in hopefully 15 years' time. I just want to say thank you to all of you, and then I should also mention a couple of people that are special, or just show them at least, and that have been my friends and my sort of peer mentors throughout my career. And there is 650 or so individuals in the various consortia I work with, so I'm not going to talk about them. But I should mention our funders, and I'm generously funded by the Lee Cushing Foundation. So thank you so much for listening. Thank you.